Hi, we're working on breast cancer diagnostics specifically for India using ctDNA. So, for our by the numbers, we wanted to first give a sort of global overview of the problem. So, for females, breast cancer was the leading cause of cancer incidence in over 157 countries and deaths in most of these countries. So, the mortality rates are pretty high. And it's estimated to be around 2 lakh newly diagnosed breast cancer cases and almost 7 lakh breast cancer related cases in 2022. And a third of these patients might have relapses, metastasis, and even may become chemotherapy resistant. So this essentially highlights the importance of looking for promising biomarkers and therapeutic agents and sort of improve early detection and therefore increase the likelihood of the treatment working. So for coming to the Indian context and why there are current problems in the breast cancer screening over here, the most common cancer type in India is breast cancer. So on an average, around 1 in 22 to 26 women have breast cancer and are diagnosed with it. However, Indians have a lower rate of survival as compared to Western countries due to the delayed initiation of treatment, which is likely due to a delayed diagnosis. So a reason why mammography doesn't work is because the breast tissue itself in Indians is a bit more dense, which sort of leads to a lot of false positives. So this may be causing too many false positives, which would further not make it the best screening tool. Additionally, there's the aspect of social stigma. There's a lack of adequate mammography machines and manpower who have the expertise to use these tools and make the correct diagnosis. And there's a skewed distribution of available resources, as in the more tier one cities or the richer places do have more access to these good quality tools and good quality healthcare and manpower, whereas the more rural areas do not. So therefore, it's also important to find an Indian specific biomarker, which is more prevalent in India, so it increases the specificity, or just one in general to make a better screening tool than mammography. So some background information about the tool that we decided to generate is given on the slide, so I'll just go over it very fast. So ctDNA stands for circulating tumor DNA, and it's present in most cancer patients, and it essentially originates from cancer tumor cells. Target ctDNA of a known sequence can be detected in biosamples, which are mainly blood and urine as part of a liquid biopsy. We are more interested in blood. And the detection of ctDNA can be performed using aptamers. The sequence of the ctDNA has to be known. Aptamers can be generated using certain techniques, which I will be coming to in the following slides. So we picked a target gene, which is the PIK3CA gene, and its studies have shown it to be found in early and late phase breast cancer patients, so easy detection. It has been, the presence of this DNA has been reported in plasma and urine. Again, it works perfectly for our tool. The target sequence for the DNA for this gene has been identified, so we already know enough about it to generate an aptamer. So aptamers are generally generated during a screening process called systematic evolution of ligands by exponential enrichment, SELEX for short. So essentially you have many single-stranded RNAs, you kind of run them through a gel and you screen the ligand receptor binding. In this case, the ligand would be our ctDNA and the receptor would be the aptamer. You generate certain KD values. So a lower KD value indicates that the binding affinity is high. And then you go on and go forth and screen and select a certain target aptamer that will work for the tool. So we have sort of thought about this, and we came up with a range for the calibration curve that we would be using for our tool. So the concentration of total ctDNA in healthy individuals is around 30 nanogram per ml of blood plasma. But in cancer patients, this goes up to 1,000 nanograms per ml. So it also could, the lower range is around 100. So we thought that 100 to 1,000 nanogram per ml should be our calibration curve for this instrument, and the level of detection, what we want, the ideal, the sensitivity should go up to 100 nanogram per ml. So again, it'll be a simple yes or no test, like a pregnancy test or a COVID test, essentially. So here is a sort of simplified design. So it's a simple lateral flow-based assay where a test and control line appear. There's a control zone that changes color always, and there's a test zone that changes color only if the result is positive. You have aptamers in your solution over here. It sort of binds to the specific ctDNA, which would then release the gold nanoparticle, and then a color is generated with certain chemical reactions. This cost is very, very low compared to what an average nanogram would be, which I think is around $250, more or less. This would be $5 max. Yeah, so this is non-invasive. It has no aspect of social stigma. It's easily accessible. It's cheaper and it's easily affordable. It could, if it's developed well enough and there are Indian specific biomarkers available, we couldn't find any. We've only found this gene. Then it could essentially build up to be more accurate than mammograms as well. It could be used in place of a mammogram to say yes or no, you might have breast cancer or any other cancer. But after this, a confirmation should be done using imaging. So it's in no way a replacement for actual imaging. So just wanted to put that out there. And we've also identified potential drawbacks of this. So blood plasma has to be separated first to be loaded onto the tool beforehand. So this can be done using a paperfuge. But again, if this tool is implemented in hospitals itself, where people have to go get the test done rather than doing it at home, it can be done in a hospital lab itself, where they have centrifuges for blood tests. So it can be done in a relatively low-cost manner. However, there might be background noise and interference from other double-stranded DNA that is found in plasma, and it's always there. We haven't sort of found a way or come up with a solution to remove that background noise. And since ctDNA is in the concentration of the nanogram range, amplification might be necessary. However, we found a low-cost method to do this. It's in a thermos, so it can be done in any environment. It doesn't need like specific temperature requirements and everything. Yeah. Future directions, we basically want to identify an aptamer. Maybe we found a company that does that, and we want to try looking for more specific biomarkers for the Indian population. This could be done by maybe looking at databases that already exist. And we would also ideally like to, way into the future, extrapolate similar systems for other cancer types. Thank you so much, and thank you to the teaching team as well.
Hello. So I have a few questions that I actually put on the chat, but I can ask directly. So this aptamer thing works with RNA, right?
It's the ctDNA. So that's the double-stranded DNA that is removed from tumor cells.
So if it is DNA, then are you looking at specific variants of this gene or just any copy of this gene?
Since it would be any copy of this gene that is there, it's found specifically in tumor cells, in breast cancer tumor cells, which is why we picked it. and it's detectable in the early as well as the late phase. So we picked this specific one. But yeah, there will definitely be issues with false positives. And as I mentioned, it could bind to essentially like other double-stranded DNA as well. If
No, so
it's
because
like two puzzle pieces. Yeah, sorry.
so the issue is that I think what you're referring to is RNA. Like when you go and look at expression, you'll see PI3 kinase being expressed a lot. But if it comes to DNA, there's only one copy per cell. So I don't know if this would work per se. And then I think people like Grail, not that this is a big problem, but they're working on this for a long time. And I don't think they've had any simple way to look at ctDNA, and they're using very sophisticated tests and charge a lot of money. They've had trouble to be able to say something about this. They use methylation, for example. Even then,
Hmm.
they're having trouble. So I don't know. I know this field a little bit. That's why I'm talking about it. I just feel like it's maybe too difficult a problem to make it into like this kind of simple frugal test. That's my worry.
Diya,
That
can you share a little bit in terms of the markers that people have looked at?
yeah.
Nobody's actually done anything on ctDNA for these specific markers as yet, right?
No, so ctDNA is being used quite a bit. Like I mean, that's the whole Grail, and there's a ton of
No,
companies.
I meant just very specifically for these breast cancer markers.
Yeah, so, but when you say PI3 kinase is a marker, that means that it's being expressed a lot, or a variant is being expressed. So either you look for the variant or you look for
The
expression itself.
RNA.
So DNA circulating is going to be just one copy per cell. I don't see how it would distinguish um, like a breast cancer from even my blood, which may not have breast cancer genes.
So there was this one paper that found that the levels of the ctDNA in breast cancer specifically were very high.
Yeah. So that's a
So
overall DNA.
yeah,
Yes. Yeah. That's different from, so just because many cells are being shed and they're dying. So what happens is often people look for these, like if you're treating, then you're killing the cells and a lot of the DNA gets spread into the blood. So you can look at the level of ctDNA totally, but there won't be any differential between PI3 kinase versus BRCA1 or P53. It's just going to be all DNA being spread out. So I'm just saying that
yeah.
this PI3 kinase DNA would not distinguish. And if you're just looking at numbers, why pick PI3 kinase? You can just look at DNA total, right? You can just look at the number of DNA molecules.
No, actually, selects can be optimized to particularly detect a particular DNA sequence. I mean, so there are companies who can do the sequence and finally generate the aptamer, which is specific to particular sequence of DNA. That is doable.
Yes, but if you're looking for variants, I agree with you. But if you're just doing DNA detection, I can do simple gel-based assays, very cheap, and you can detect DNA of particular genes or particular sequences. So I'm just saying there's a little bit of a technological gap here that has to be bridged.
Yeah. Ravi, let's do this. Let's
Yeah,
do
sorry.
a catch up on the
Yeah,
same. I just want to
yeah.
make sure everybody gets to finish.
Sorry. Yeah,
Yeah.
I didn't mean to monopolize. Sorry.
No. I think it's very important because the whole point of this is that this is sort of the initial steps to make sure that we do figure. I mean, I think there is a general value in thinking about floating DNA. So I think overall, even if the current set of a framework is hard, it would actually be really valuable to have that discussion.
Yeah,
So
yeah.
Diya, we should definitely just post on Discord a little bit of a time.
Oh, yeah. And I have some
discussion on
ideas
this.
on mitochondrial DNA. I can show you some data. Yeah, it'll be
Yeah,
fun. Yeah, let's
yeah.
do that. Yeah.
Let's do that as
Yeah.
a follow-up.